welcome you all to today's video. Today is the next video of the video series where we are talking about concepts of genetics which will help class 12 students as well as medical aspirants. In the last video, I have talked about law of dominance and law of segregation. Today we will see what is law of independent assortment, as well as we will see the cases and concepts where these laws of inheritance are not applicable, as well as the ratios which are obtained by crossing individuals in these cases are different from that of monohybrid and dihybrid crosses. These cases are studied under heading post-Mendelism. These are codominance, incomplete dominance, pleiotropy, multiple allele, polygenic inheritance, epistasis, lethal allele. I have already uploaded videos on individual these topics with examples and concepts which are given in the playlist. Today I will just discuss briefly about these four things, as these four things are given in the book NCERT along with examples. A dihybrid cross involves two characters, that is two gene pairs are involved over here. This is one gene pair, this is another gene pair. This is one allele pair, this is another allele pair. In case of monohybrid cross, only one gene pair or one allele pair was involved. So over here, the yellow and round seed containing plant is crossed with a green and wrinkled seed containing plant. The gene for yellow and round are present on different chromosomes, that is 4 chromosomes are involved over here. This was the thing which made Mendel so successful. I will explain its importance in my next video. That is, the allele capital Y, suppose they are present in chromosome number 1 and 5. So capital R will be present on chromosome suppose 6 and 7. This made Mendel so successful. According to law of segregation, every allele pair will separate from one another. That is, when gametes will be formed, one Y will enter into one gamete from this pair. Similarly, one R will enter from this pair into the same gamete. So suppose first gamete is having capital Y, capital R. So this capital R, Y has moved with this capital R. In another gamete, this capital Y will and this capital R will enter. So every gamete will have at least one allele according to law of segregation. According to law of independent assortment, this capital Y can move with this capital R, or this capital Y can move with this capital R, because these are only 4 chromosomes. There are 14 chromosomes over here, and every chromosome is having many genes. When gamete will be produced, 7 chromosomes will enter into one, another 7 will enter into another, according to meiosis. So when gamete will be formed, when chromosomes will move, they will move independent to each other. That is, there is no fixed pattern. That is, capital Y can move with capital R or this capital R. It is not fixed that only capital Y will move with this capital R. There is no fixed pattern. This is called as law of independent assortment. That is, the gametes are formed when the alleles can assort, that is mix independently. This capital Y can move with this capital R, or this capital Y can move with this capital R. Any probability can be there. For example, there is one genotype, capital Y, capital R, small r. As you all know, they are present on different chromosomes. So when gametes will be produced, this capital Y can move with this capital R, or this capital Y can move with this small r. So you can have gametes with capital Y small y capital R, and small y small r. So 4 types of gametes will be produced because of law of independent assortment. And this was possible in every generation because the genes which he had selected were present on different chromosomes. What would have happened if they were present on same chromosome? That is, let's suppose capital Y and capital R are present on same chromosome. I will answer this question in my next video. The most important contribution by Mendel was the concept of dominance. To understand dominance as well as the cases of post-Mendelism, that is codominance and incomplete dominance, let's take one example. Over here, the 2 flower colors are chosen, that is, a red flower colored plant is crossed with a white colored flower plant. A1 is the allele that produces red pigment and A2 is the allele that produces white pigment, because of which the flower having genotype A1 is red in color and genotype A2 produces white phenotype. A1 is dominant over A2, which means when A1 and A2 will be present, only red pigment will be produced, as a result of which the flower will be red in color. So when red and white homozygous flowers were crossed, the F1 which was produced was a heterozygote with genotype A1, A2. This is red in color because A1 is dominant over A2. When selfing was done, A1, A2 was crossed with A1, A2, that is, this is F1. When F1 was selfed, these are the F2 offsprings. A1, A2 are the gametes produced from this parent, and A1, A2 are the gametes produced from this parent, and these are the F2 individuals. This is A1, A1, this is A1, A2, this is A1, A2, this is A2, A2. This is red in color because A1 is present in homozygous condition. These two are also red in color because A1 is present in single copy and A1 is dominant, and this is white in color. So when dominance is the concept, then we will get the ratio of 3:1, 3 that is red and 1 is white in color. This is the phenotypic ratio, and genotypic ratio is 1:2:1. 1 is A1, A1, and this one is A2, A2, and this 2 is A1, A2, A1, A2. These are 2 in number, this is 1 in number, and this is 1 in number. In case of incomplete dominance, any one allele cannot completely dominate the other allele in case of heterozygote. So over here the heterozygote will not be red in color because A1 allele will not produce sufficient amount of red pigment to cause that flower red in color. Suppose in case of dominance, A1 produced 10 units of pigments or 10 molecules of pigments, but in case of incomplete dominance, the same A1 produces, suppose let's see 8 units of or 8 molecules of pigments. So in case of heterozygotes, in case of incomplete dominance, the units of pigments produced by A1 allele is insufficient. That is why the heterozygote flower will not be red in color. It will be somewhat lighter in shade than red. So let's suppose this F1 is pink in color, and this pink is of any shade, that is light pink, dark pink, or very dark pink, any shade it can be. So this heterozygote is pink in color. When selfing was done, that is one pink flower was crossed with another pink flower from F1 generation itself. These are the F2 offsprings, and F2 offsprings, this is red in color because there are 2 copies of A1, and these both are pink in color because these both are heterozygotes, and this is white in color. So the phenotypic ratio over here is 1:2:1. 1 is red and this one is white, and this 2 is pink. So these 2 individuals are pink in color. The genotypic ratio is also 1:2:1. 1 is A1, A1, and this one is A2, A2, and these 2 are 2 in number. So this is represented by this 2. So in case of incomplete dominance, the genotypic phenotypic ratio is the same. This happens because the heterozygote over here is an intermediate of both the homozygotes. This homozygote is red in color, this homozygote is white in color, and this heterozygote is an intermediate. Intermediate does not mean that the color will be exactly in between red and white. It can be of any shade. In case of dominance, the heterozygote will be similar to any of the homozygote. Over here, this heterozygote is similar in phenotype to this homozygote. In case of incomplete dominance, this heterozygote is intermediate to the phenotypic expression of both the homozygotes. In case of codominance, both the alleles produce equal amount of pigment. So the heterozygote will have both the shades, that is, the petals of that flower will have red color, then white shade, then red color, then white shade. So when this red and pink flower will be crossed with itself, that is F2 F1 individuals, when they will be selfed, we will get the F2 individuals. This one will be red because this is an homozygote. These 2 will have both the colors in their petals, that is, these both will be red and white in color, and this one will be white in color. So in case of codominance, the heterozygote will include both the homozygotes. See the difference. In case of dominance, the heterozygote is similar to any one of the homozygote. In case of incomplete dominance, the heterozygote is intermediate to both the homozygotes. But in case of codominance, the heterozygote includes both the homozygotes. That is the phenotypes. We are talking about the phenotypes. So in case of codominance also, the genotypic ratio will be 1:2:1 and phenotype will also be 1:2:1. One stands for red, the another one stands for white, and these two stands for both red and white flowers. Let me tell you, this white color in natural cases is due to lack of pigment. As you all know, white is not a color, so it is due to lack of pigment. But for an example or for an easy understanding, I have taken one hypothetical example so that I can explain you all the three cases. Naturally, the example of incomplete dominance is the flower color in case of snapdragon, that is both red and white. The same example is used to explain incomplete dominance. And the example of codominance is blood group. The example of codominance is blood group. In case of human beings, the most important blood group system is ABO system. This ABO system is due to the presence of carbohydrates on the plasma membrane of RBCs in blood of human beings. So suppose this is one RBC, and this the plasma membrane of RBC has many molecules on its surface. One of them is a type of carbohydrate. So the individuals having a type of carbohydrate which is named as A, so those individuals are said to have blood group A. The individuals which have B blood group have B type of carbohydrate on them. There is a structural difference between A and B, and they are named on the basis of the structure. The individuals having AB blood group have both the carbohydrates, that is A type and B type, and the individuals which have blood group O have the absence of both of these carbohydrates on their RBC. These carbohydrates are synthesized by the help of a pathway in which one enzyme is required, and that enzyme is synthesized by the help of a gene. That gene is called as the blood group gene, and that gene is having three alleles, that is IA, IB and small I. The gene that helps in determining plant height is having allele 2 types, that is capital T and small t. But in case of blood group, the gene is having 3 types of alleles. So blood group is also an example of multiple allele, because there are many cases in human population or in animals or in plants also where one gene is having more than 2 alleles, but each individual is having only 2 alleles. In population there are 3 alleles. So every individual can have any 2 alleles from this group at one locus. So suppose that gene is present on this chromosome. So these are the 2 loci. These are the 2 locus where that alleles will be present. So in every individual, any 2 alleles will be present from these 3 alleles. IA allele helps in production of the enzyme that helps in production of A type of carbohydrate. IB allele helps in production of enzyme that helps in production of B type of carbohydrate. Whereas small I does not produce any enzyme, as a result of which no carbohydrate is produced. And IA and IB are dominant over small I. So the individual which has genotype IAIB will have blood group AB.
The individual with genotype IAIA or IA small I has blood group A, because only one type of enzyme will be produced to form one type of carbohydrate, that is carbohydrate of A type. In blood group B, the genotype is IB, IB or IB small I. So B type of enzyme will be produced to form B type of carbohydrate. In blood group O, the genotype will be small I, small I. Small I is the non-functional form of the allele for the gene which codes or which works for blood group of human beings. And this small I cannot form any type of enzyme, and thus any type of carbohydrate is not formed. So the blood group is O type.
The next case of post-Mendelism required for class 12 students, which is given in NCERT, is that of pleiotropy. The meaning of pleiotropy is when one gene controls more than one phenotype. Normally a single gene controls a single phenotype. Suppose flower color is controlled by a single gene and that gene does not affect any other phenotype. But there are many cases where one gene is seen to control more than one phenotype. The example which is given in NCERT is a gene that is responsible for starch synthesis in case of seeds, and that gene is having allele capital B and small b. The homozygous individuals, that is capital B, have good amount of starch production because there are two alleles which help in production of starch, as capital B helps in production and small b cannot produce starch. So these individuals have round seeds and good starch production. Small b individuals have less amount of starch production, that is no starch production, and these seeds are wrinkled. The intermediate genotype, that is capital B small b, is having intermediate starch production, as a result of which the size is also intermediate. So over here the same gene, that is gene for starch synthesis, is controlling two phenotypes, that is starch production as well as seed shape. This case can also be seen as an example of incomplete dominance because this heterozygote is an intermediate to these both homozygotes. So this is also an example of incomplete dominance. With this we come to the end of this video. In the next video lecture we will talk about linkage and recombination as well as gene mapping. We will also solve few problems on gene mapping. The definitions of the concepts discussed are given below in the description box, as well as the links of the videos which are already uploaded in genetics are also given in the description box. Thank you for watching.